Thank you, everyone. Thank you very much to the Jung Foundation for inviting me to talk here today, for this great opportunity to present our work. I'm going to talk about personalized immunotherapy, how we can develop the best cancer treatment for every patient. And I added something by targeting the unconventionals. So a brief introduction on cancer immunotherapy. As I think most of you are well aware, cancer immunotherapy has really revolutionized the way how we treat cancer. Employing the body's own immune system to target the immune system against cancer has been a major breakthrough, and it has been acknowledged in many ways. Has been a science breakthrough of the year 2013 and also led to the Nobel Prize in Medicine and Physiology for James Allison and Tetsuko Honjo. So it has also entered the main stage for treating liver cancer. And what you can see here is the two major phase 3 clinical trials that we have for advanced liver cancer. And what you can see here is that the combination immunotherapy has been a major benefit for these patients compared to previous standard of care. Has really prolonged life for many of the patients with advanced liver cancer, with advanced HCC. But what you can also see here is that the prognosis is still poor. What we see and what we want to see is an immunotherapy that has an effect like this. And what keeps me up at night is how can we get there? How can we improve immunotherapy for patients with liver cancer? So the approach that we have in our group now in Tübingen is the following. As a clinician scientist, I work in the hospital and I see patients with liver cancer every day. And I can see, unfortunately, for many of these patients, we don't have the best treatment options yet. So there's a big unmet clinical need. And what we do is we collect patient samples, the tissue from the organ where the tumor arises, the tumor tissue, blood, stool samples, and we try to analyze these samples in a very comprehensive way, try to get as much information as we can from these clinical samples. Try to do an integrated analysis to come up with hypotheses why an immunotherapy is working or why it's not working, and what's the difference between the patients, because so far we don't have good ways to stratify these patients and identify biomarkers for which immunotherapy would be the best for each patient. Once we've come up with a hypothesis, we then go back in the lab and do functional studies using patient samples and in vivo models. And obviously, as a clinician scientist, my dream would be to translate this back into the clinic and try and test these approaches in clinical trials. So I've previously introduced our work on the tumor microenvironment. We try to understand the tumor not only from the tumor side, but we also try to see what's happening around the tumor, because the interplay between the tumor cells, stromal cells, immune cells, has major impact on cancer biology, but also on how a patient responds to immunotherapy. And we study the liver because I think from an immunological point of view, the liver is a fascinating organ to study. It's a big immunological organ, and we can identify some immune cells that we don't have anywhere else in the body, such as the tissue-resident macrophages, the Kupffer cells. But the liver is also home to a lot of tissue-resident lymphocytes. They're patrolling the liver sinusoids and they protect us from intruding pathogens. So I think the liver is a very special organ. And the question now here is how can we identify novel targets for immunotherapy of HCC? And I want to introduce some of the work that led to this award here today. So what we did here is we had a cohort of patients with liver cancer and we collected tissue specimen. We collected adjacent liver tissue from the tumor border, but also from the tumor itself. And we tried to do this comprehensive analysis where we separated some tissue for in situ high-plex immunofluorescence imaging, but I also generated single-cell suspensions for high-dimensional profiling of the immune cells in suspension. And this is where I want to start. So when you take the immune cells out of these cancer patients, liver cancer patients, you generate a single-cell suspension and suspect these cells to single-cell RNA sequencing, then you can generate an immune cell atlas of liver cancer, if you want to say so. So in this UMAP projection, you can see how cells are clustered according to their transcriptional similarity. So each dot in the plot is one cell, and the more similar these cells are in the transcriptomic profile, the more closely they cluster together. And with this approach, you can identify key marker genes to identify all the immune cell subsets in the liver and in the tumor. Using these marker genes, you can identify B cells, T cells, innate immune cells such as NK cells, but also myeloid cells such as monocytes or macrophages. If you look at this plot down here, you can see that all of the patients that we looked at contributed to each of these immune cell clusters in a bit heterogeneous way. And you can also see, if you look at where these cells were coming from originally, was it the adjacent liver, the rim, or the tumor core? So green is the tumor core and red is the adjacent liver. There seems to be some preference that some of the immune cell clusters were coming mainly from the tumor or mainly from outside. And this is what we looked at here. So in red, you can see the frequency of the cells in the adjacent liver, and in green will be the ones that are coming from the tumor core. And we found some interesting patterns. If you look at these cells that seem to be excluded from the tumor, they don't make it inside the tumor. It's potent killer cells such as natural killer cells, but also CD8 T cells that express coenzyme K, which we know these cells are immune cells that can contribute to anti-tumor immunity. So these cells somehow don't make it inside the tumor. And that was something that really caught our attention. And another cell type that was also following this trend was the mate cell cluster. And then I was like, what are mate cells? So mate cells, very abundant T cell subset in the human liver, can make up to 30 to 40, 50% sometimes in the healthy liver as a frequency of T cells. And these cells belong to the compartment of innate, like, or unconventional T cells. They're unconventional because somehow they share properties of both innate immune cells but also adaptive immune cells. So in this graph here, you can see the innate immune cells on the left side, NK cells and innate lymphoid cells. On the right side, you can see our adaptive immune system represented here by CD4 and CD8 T cells. And these CD4 and CD8 T cells have T cell receptors that recognize peptide ligands presented on MHC class 1 or MHC class 2. The group of innate-like or unconventional T cells share properties of both of these cells, the best of both worlds. They have also a T cell receptor, and mate cells with their T cell receptor, they recognize riboflavin metabolites that are presented not on MHC class 1 or MHC class 2, but an antigen-presenting molecule called MR1. And they can rapidly produce a lot of cytokines, and that is why we think they might be a good target for immunotherapy. So in the context of the physiological role, these cells recognize these bacterial metabolites. The metabolites are presented on MR1 through the T cell receptor. Mate cells get activated, they proliferate, they can form a rapid cytotoxic response by producing perforin, granzymes. They can recruit other immune cells such through signaling like IL-17 or interferon gamma or TNF. But not only can we activate them in a T cell receptor dependent way, but we can also activate them independently. So through two separate pathways, through the high expression of cytokines, for instance. So we're basically here at this stage, hypothesis generation. So the question here was, what are mate cells and what are they doing in HCC? They follow this interesting pattern, such as the NK cells and cytotoxic CD8 T cells. So what is their role really in HCC? What we first did is we looked at published data sets. You can see here TCGA, LCI, and Thai-HCC published RNA sequencing data sets. And we stratified the patients in these published data sets by signature, mate high versus mate low. So the blue line, mate high, seemed to correlate with better overall survival in these HCC patients. So that was intriguing. Could mates have protective role against liver cancer? So to have a bit more functional idea of these cells, we went to our mouse models of HCC, where we have fully immunocompetent mice, and we can implant syngenic liver cancer cells into the liver of these mice. And I tested two different mouse strains, one that has mates and one that doesn't have mates. So black 6 mice are the wild type, and if you knock out the antigen-presenting molecule on the mouse germline, then these mice never develop mates. So in the wild type mice, you have a small population of mate cells in the liver, and that's a big difference. In human, we have lots of mates. In mice, it's quite a small population. But nonetheless, if you look at the knockout mice, you find a mouse strain that lacks this immune cell type completely. And then if you look at the tumor growth at the end of the experiments, you can see wild-type mice have smaller tumors that are growing in the liver compared to the MR1 deficient mice. Significantly higher tumor burden in these mice that lack mates. So this kind of suggested that mate cells can have anti-tumor properties, because if you take them away, the tumors are bigger. And then the question was, what's happening to these cells in our HCC patients? Again, back to our study, and again back to our single-cell RNA sequencing and flow cytometry experiments. We expanded the cohort a little bit and collected some more patient samples, and this was confirmed here, that we see that mate cells are excluded from HCC tumors. They're somehow stuck outside the liver. And if you look at the transcriptomic profile in HCC, you can really zoom in into the subsets of mates. And this is what we did here. We took the mate cell clusters, subclustered these cells, and what you find is that some of these cells look like they express genes that indicate dysfunction in the context of tumor, and they lose their cytotoxicity. So dysfunction indicated by CTLA4 expression, PD-1 expression, loss of cytotoxicity by a lower expression of these effector molecules such as granzymes and perforin, interferon gamma. So our conclusion here for the first part was, first, we think that mate cells could be potentially an interesting target for immunotherapy of HCC. We've seen that mates seem to play a role in the tumor growth of liver cancer in mice. And what we found in our patient samples is that they seem to lose their cytotoxicity and they don't make it into the tumor. And the question here was like, what are the mechanisms? What stops these cells in our patient samples from infiltrating the tumors? What might be cellular interactions in the tumor microenvironment that are detrimental for mate cells in HCC? And then back to our study, where we used our tissue collection from the same patient samples that we also collected the cells from. And what we did is we tried to generate a spatially resolved immune cell atlas of HCC. And what you can see here is that we used a technology called CODEX, which is basically a high-dimensional immunofluorescence assay, and we stained large tissue sections. What you can see here is an overview where you can see that there's a big heterogeneity in the tissue, adjacent liver, tumor margin, tumor core, and we profiled these samples with an antibody panel with 37 markers that allowed us to study both parenchymal cells, tumor cells, but also the immune cells, and talk about functional status of immune cells in the intact tissue context. And this is really a powerful technique and a really powerful technology to study the heterogeneity of the tissue, right? Because all of the patients receive the same treatment, but what we don't appreciate enough is how heterogeneous the tumor microenvironment, and each patient really is. So if you this is just one patient sample. You see here the adjacent liver tissue, very different than the tumor, very different also to this peritumoral region. In the adjacent liver, you have the liver sinusoids, you have the patrolling immune cells in the sinusoids. Here is a portal region where the blood from the gut through the portal vein reaches the liver, so you have a lot of immune cells around these vessels. You can find the tumor. There's a lot of neovasculature in the tumor that's not supposed to be there. You have proliferating tumor cells, and you have around the tumor this big stromal region, but you also find regions where you have a really coordinated immune response against the tumor when they form tertiary lymphoid structures, where basically B-cell responses are generated right outside of the tumor. So I want you to dive into the tissue with me. And what I love about these tissues is how much you can learn about the tissue by visualizing it. So we're taking a deep dive into the adjacent liver now. And we were zooming into a region where we have the portal triad with the artery, portal vein, and the bile duct area. And you see here all the liver sinusoids that are entering into the liver vein here. And we can use this technology to look at all the different aspects of tissue biology and the structural organization of liver tissue and liver cancer as well. So just an example. So if you look at all the immune cells, you can see the heterogeneity already in this small tissue section, where we have a lot of immune cells around the vessels up here, also around the portal, the liver vein here. But you can also see that some of the immune cells are scattered in the liver sinusoids, which you can see here. When you turn on the markers for the liver sinusoids, you see that these cells are patrolling the liver sinusoids. And now we can try and see what are these cells now. Macrophages. The yellow one, CD68, highlights the macrophages, tissue-resident Kupffer cells sitting in the sinusoids, eating all the bacteria that invade our body through the portal vein. We can look at T cells, our adaptive immune cells, and we can see how they're located next to the vessels here. But we can also see many, many examples where these T cells interact with macrophages in the liver sinusoids. But it becomes more complex than just that. Granulocytes, most abundant immune cell type in the human body. We have B cells. And that really just gives you an example on how complex these tissues are and how important it is to understand the complexity of the tissue when we want to really improve patient care for patients with HCC. And why we think these assays are useful and meaningful, because it allows us to not only talk about the different immune cell abundance, how many T cells are inside, outside of the tumor. We can talk about how dense they are, what's the density of the cells in the tissue, what phenotypic markers do they have in the different regions of the tissue. Are there maybe spatially restricted expression patterns? What are the cellular co-localizations? Which cells sit next to each other, which might indicate that they interact with each other? And what is the distance between the different cell types? So this becomes really, really complex really fast. So you need good partners to help you analyze these complex data sets. And we teamed up with a group of bioinformaticians at the University of Tübingen, a group of Manfred Klaassen. They developed a machine learning algorithm to trying to identify the higher order tissue organization in these HCC samples. And I'm not the best person to explain how this works, but basically what they did is they trained a convolutional neural network to generate a filter and see what cells are preferentially enriched in the mate cell niche in HCC. So what are the cells sitting around mate cells in these HCC patient tissues? And this plot I like more because this is easier to understand, because the highest enrichment score indicates the most enriched cell type in the mate cell niche in HCC. And those were tumor-associated macrophages that express PD-L1. So we found that these potentially immunosuppressive cells are really enriched next to the mate cells in the tissue. And while this is not so easy to understand and not so easy to explain, what I love about these images is that you can always go back. You can always go back to the primary image and you can see how the mate cell that expresses the T cell receptor and the mate cell that also expresses PD-1 is really closely interacting with these macrophages expressing CD163 and also PD-L1. So the interaction between PD-1 on mate cells and PD-L1 on macrophages here really gave us an idea about what's happening, why these cells could be impaired in liver tissue. So now we're at the stage of the validation experiments. And what we did here and what I want to highlight here is one experiment where we collected some more liver tissue. We isolated the immune cells and sorted both macrophages and mate cells, co-culture them together, and measure mate cell function with cytokine analysis at the end of the experiment. And what you can see here is that the mate cell function as measured by interferon gamma really decreases if you add more macrophages to the co-culture system, but if you take non-macrophages, this really doesn't happen at all. Another question was, is this really a cell contact dependent mechanism, as we saw in the tissue where the cells are really sitting closely next to each other? So if you now physically separate these cells with a transfer system, you can study the effect of this cell contact. And what you can see, if you separate the cells, the macrophages from the mate cells, then the mate cell function is partly restored. They produce more interferon gamma. And now the question was, is this really PD-L1 mediated? Then you can add a blocking antibody to this co-culture system, and you can also see if you block the PD-L1, then you can reverse the mate cell dysfunction, at least partly. So at this point, the conclusion was that these PD-L1 positive tumor-associated macrophages, they trigger mate cell dysfunction at the HCC invasive margin. We can reverse this in vitro and in vivo system. But what this also suggests, that all the patients that we treat with PD-1, PD-L1 in the clinic, the mate cells might also be a target cell, because this pathway clearly seems to be affecting these cells in liver cancer. Okay, in the end, we found PD-1, PD-L1. Great. But can we also target these cells more selectively? Can we target them more specifically for cancer immunotherapy? And this is what we're still working on in the lab, in collaboration with the chemist who helps us synthesize mate cell ligands. These riboflavin-derived T-cell receptor ligands for mate cells. In a very simple experiment in the lab, you can add these ligands to PBMCs, and you can see that the mate cells express higher levels of interferon gamma for some of these ligands. Some are not as activating. So what we did, we took the lead candidate, 5-OPRU, and studied the function of 5-OPRU in the context of tumor control for mate cells. So the experiment here again, we have mice with a syngeneic liver cancer model, and we can inject these mice with this T-cell receptor ligand called 5-OPRU in combination with an adjuvant, CpG, twice a week, and then check out what happens at the end of the experiment. And what you can see here is that this small population of mate cells really expands 20, 30, 40, 50-fold in the liver of these mice. And what happens to the tumor? They shrink. This is now a melanoma metastasis model where the melanoma cells were injected into the liver. So this black here is unfortunately a big tumor for the mouse. But if you apply the 5-OPRU CpG treatment to these mice, tumors are smaller, and also these mice can live longer. And at this point, we were wondering, okay, we think mate cells are potent mediators of an anti-tumor immune response, but are they really the killers? Are they really the effector cells? Or might other cells be also involved in this effect? So what we did is an immune profiling of these mice. And not only mates expanded in these mice, but also CD8 T cells. What you see here is when you treat the mice with 5-OPRU and CpG, you get a higher infiltration of CD8 T cells in the tumors. You can study antigen-specific T cells using these tetramers, and you can see that also the antigen-specific CD8 T cells infiltrate these tumors more, which correlates with the size of the tumor. And I talked about mates being in between adaptive and innate immunity. We also studied these innate immune cells, so T-cell beta negative NK1.1 positive cells in the mouse liver. Those are NK cells and helper ILC1s. And what you can see here is also those cells infiltrate tumors more. And not only do they infiltrate tumors more, they also increase their cytotoxicity, expressing more perforin. And now, can we use this treatment to overcome resistance of immunotherapy, right? Because I started this talk introducing like how can we identify the best immunotherapy for each cancer patients. We identified mate cells as a potential target, and we've seen the clinical need that some patients don't respond to immune checkpoint blockade, so that's what I tested. And again, I tested wild-type mice that have mates or mice that don't have mates. Injected the tumor into the mouse liver and then treated these mice with immune checkpoint blockade using anti-PD-L1 treatment and or the mate cell treatment. And what you see in the wild-type mice, so here I chose a dosage of both the PD-L1 and the 5-OPRU that didn't really control tumor, but if you combine the two treatments, anti-PD-L1 and mate-directed immunotherapy using 5-OPRU and CpG, then you can control the tumor, and you really need mate cells for this because it doesn't work in the MR1 knockout mice. So to conclude my talk here is the last conclusion. identified mate cells as a target for immunotherapy, and we can see that these cells can mediate an anti-tumor immune response in mouse models of HCC, and we need mate cells for that. Mate cells subsequently activate CD8 and NK cells and could potentially overcome resistance to immunotherapy, at least in these mouse models. And what I think what the data tells us here is that mate cells really bridge innate and adaptive immunity. And with that, the outlook would be that, of course, as a clinician scientist, at one point I wanted to retranslate these findings into clinical trial, and that's also one part why the Jung Foundation supported this project. And I'm really, really grateful to be here today. Thank you again for all the members of the Jung Foundation, the scientific committee, and all the people who worked on these really interesting projects. I think I had so much support throughout my career. My mentors have always supported me, all the people in the lab now who are working hard on this. And thank you so much for having me today. Vielen Dank.